Morning all. Last class we completed endonucleases enzymes. Actually, we started introduction about endonuclease enzyme. Today we discuss about nucleases enzyme, restriction endonucleases enzyme, ligase enzymes, phosphatase enzyme. There are. First, let's discuss about restriction endonucleases. They are mainly used in genetic engineering process. Right. Restriction endonucleases. The restriction enzyme, it is a nucleases enzyme. The nuclease enzyme function I already told you, that is cleave. Which is nuclease enzyme that cleaves the DNA sequence. That is the function of this nuclease enzyme. That cleaves at random site, random of the DNA or specific recognition site. Maybe it is random or specific site. That is known as restriction site. So the work on restriction site, that is called as a restriction enzymes. In bacteria, restriction enzymes form a combined restriction and modification system. With the modification system enzyme methylate, I will explain later, methylate bacterial DNA methylation process. There are two kinds of restriction enzymes. One is exo, another one is endo. The first, let's see the exo. Exo, which is catalyze the hydrolysis of terminal nucleotide from DNA or RNA molecules 5 to 3' direction or 3 to 5' direction. So for example, this is 5' to 3' 3 prime, always opposite direction. Here the 5 prime to 3 prime. So terminal nucleotide, for example, here the only terminal nucleotide. What is nucleotide? Nucleotide, nitrogen base, sugar, phosphate linkage. This is called nucleotide. So this terminal side, whether it is 3 prime or 5 prime, with help of the water, separated. With help of water separated. So that is the exonuclease. Outside that mean endo, any ending. Endonuclease can recognize the specific base sequence restriction site within DNA, not terminal. Exonuclease and terminal site. But within DNA, internal, within DNA or RNA molecule, and cleave the internal phosphodiester bonds. Here the terminal. But if you take the structure of this sugar, nitrogen bases, at the double bond, thymine, again sugar, phosphate. Here also same diphosphate group, and again sugar and phosphate, sugar, phosphate. So sugar diphosphate, always sugar diphosphate. Here thymine, guanine, thymine opposite adenine, guanine opposite cytosine. If the terminal act, for example, this terminal hydrolase act, that is the exonuclease, but internal, in middle cleave internal phosphodiester. This one, cleave this internal phosphodiester. If it is cleave the internal phosphodiester bonds within DNA molecule, that is called endo. Within the DNA, in middle, that is called endo. The example, Escherichia coli, restriction endonuclease 1, into 3, bomb. They are the different type of restriction endonuclease enzymes, right? And let's see some history. In 1970, first restriction endonuclease enzyme into 2. First restriction endonuclease enzyme was into 2, was isolated. For the subsequent discovery of characters numerous restriction endonuclease, Daniel Nathan, 1978. Daniel Nathan, Werner Arber, and Hamilton O. Smith, awarded for Nobel Prize for Physiology Medicines. Nathan, Werner, Hamilton. Since then, restriction enzymes have been used as an essential tool in recombination DNA technology. So these are all enzymes used for DNA technology, recombinant DNA technology to cleave the DNA fragment, desirable DNA. To get desirable DNA, we can use restriction endonucleases. So first discovered endonuclease into two, that is type of restriction endonuclease by the Daniel Nathan, Warner Harbour, Hamilton. Restriction endonuclease nomenclature. Of course, here one nomenclature in book, another nomenclature given. But anyway, anyone you can write anyone, not a problem. In our book, Eco R1, but here into 3. So here restriction endonuclease. Restriction endonuclease are named according to organism which they were discovered using system letter numbers. Example here, one of the example, the first into 2, actually, the second into 3, pronounced in D3, was discovered in Haemophilus influenza. Haemophilus influenza. The Roman numericals are used to identify specific enzyme from bacteria that contain multiple restriction enzyme, including in order in which restriction enzyme were discovered in particular strain. Here, for example, hind 3, first letter H. H, which is this letter genus name, represents the genus name Haemophilus, H for Haemophilus, H Haemophilus. And first two letters, species name influenza, IN influenza. This is H, genus name, IN ind. Influenza, species name. D. D is the strain type of strain T. 3, for the third enzyme discovered in that organism. Of course, already told you, and if you take any organism, there are number of enzymes there. The based on the number, we can give that. First enzyme 1, second enzyme 2, third, fourth, fifth, like this. Send. In our book, given the page number 106, here the one nomenclature type, the example, Escherichia coli, E, O, R, I, the given. So in this, E abbreviated, E for, already told you, genus name, Escherichia. CO, callus name, species name. R, type, strain type, R. strain type R. And first, that is the number of the sequence. First time recognition, first engine, that is called given first. So this is about restriction endonucleases nomenclature. Nomenclature also not an important one. Classification important. Classification restriction endonucleases. So the classification restriction in endonucleases, they are major classes endonuclease based on the types of sequencing are recognized. Based on sequencing recognized, this is important. Based on sequence recognized the nature of cut and also nature of cut made in the DNA enzyme structure and also dual function. I will explain later. Dual function means endonucleases activity and missionase activity. So based on that, we can classify the restriction endonucleases three types generally. Restriction type 1, type 2 endonucleases, and type 3. First, let's see the type 1 endonucleases. So type 1, this is important here. This enzyme have the both restrictions that I already told you based on the restriction side, restriction and modification activity. One enzyme, only one enzyme, two activities. Can modify methylation, restriction recognition. So restriction depends upon the methylation status of the target DNA. So cleavage occur approximately here 1,000 base pairs away from the recognition site, but not exactly recognition site. First, it recognizes the recognition site. After that, for example, this is the DNA strand. Here recognition site, but cleave occur here in between 1,000 base pair above. So the recognition site is asymmetrical and composed two specific portion. One portion contain 3 to 4 nucleotides, while other 4 to 5 nucleotides. And they require for this activity, restriction and endonuclease activity, it require S adenosine thymine methionine ATP energy magnesium for the activity. These enzymes are composed mainly three subunits, specific subunits that determine the DNA recognition site. The subunits are the subunit, maybe the three subunits are okay. The three subunits are HS, it is specific subunit determine DNA recognition site. Three subunits here. The three subunits are HSDR, HSDM, HSDS, DM, DRS. So HSDR, R itself, restriction, M, methyl group, M for methyl group, modification. And S is important specific cut recognition addition methyltransferase activity. So this is the three important subunits. And if you take the restriction endonuclease 2, restriction and modification mediated by separate enzyme, but not same. That is the first point. If you take the restriction one enzyme, restriction and modification same enzyme, but here mediated by separate enzyme. So it is possible to cleave DNA in the absence of modification. But in first one, the modification takes consideration first mainly. All the two enzymes recognize the same target sequence by two enzymes. One enzyme for one enzyme for restriction, another enzyme for methylase activity, and they can purify separately from each other. The cleavage nucleotide sequence occur at the restriction site, but not away from the 1000, 2000, 3000. Exactly, cleave at the cleavage site. This is the DNA, it is cleave site. Here, restriction. When the cleavage takes place. The enzymes are used recognized rotationally symmetrical sequence, which is often referred as palindromic sequence. That restriction should have some palindromic sequences. I will explain the palindromic sequences later. Here, the palindromic sequence may be G, G, 5 to 3, and G, T, T, N, E, C. GGTACC. So for this activity required only Mg2 plus as a cofactor. ATP and SCAP, they are not involved in this. So the type 2 endonuclease widely used in mapping and reconstruction DNA in vitro because the recognized specific site cleave just these sites. So that's why actually this restriction to enzymes called as work horses in this because it is mainly used in different type of technique. That's why it is called as work horses. Remember. So restriction endonuclease called as work horses. Restriction endonuclease 2. The steps involved in DNA binding cleave by type 2 restriction endonucleases. First of all, these enzymes have the non-specific contact with the DNA, initially bind to DNA as a dimer. As a dimer, the target site then located by combination linear distribution sliding of the enzyme along the DNA over short distance. Uh, Here, let's see. So this is DNA. So DNA stand restriction endonuclease to this one, and it is recognized. Actually, this is non-specific binding sliding, and actually this is the specific site. This is recognized site, but the bind just below the recognition site. But cleavage occurs this restriction site. How it is cleave? How the cleave here? Let's see. So yellow color restriction endonuclease 2. Now it is cut exactly here. G in G. Different sites, but name different site. Now it is separated as now these are called sticky ends because this is ready to stick another stand. Here also ready to stick another stand. These are also ready to stick another stand. Here also ready to stick another stand. That's why these are called sticky end. The two dimers are shown in brown and DNA, back brown, green, which no need to write all this. Restriction 3. These are recognizes methylase same like a first one. Recognition sequence same DNA sequence, but cleave 2 base pair away. 24, 26 base pair away. They have two different subunit. M responsible for recognition, modification DNA. Other R for nuclease action. Magnesium 2 plus ATP are needed for DNA cleavage, but not SAM. Cleave stimulated by SAM. The cleave only one stand. To recognize site in opposite orientation are necessary bracket duplex. Opposite, directly opposite. Like for example, this one, cut like this. Due to this, blunt ends are formed. In first, sticky ends formed, later blunt ends formed due to opposite cut. Here given some of the comparison property abundance. Which enzyme is abundance in nature? The type 2, type 1, type 2, type 3. This is less common than type 2. Most common, rare. So in this first position in abundance, 2 is the first is the second, third last. Recognition site. Cut the both stand at a non-specific location away 1000 base pair away. Cut the both the stand at specific, non-specific. That is random, already told you random, but here specific. The specific sequence called as palindromic. Recognition site 4 to 8 base pair nucleotides. Cleavage of any stand only 24, 26 base pair downstream of this recognition site. Restriction and modification. Single multifunctional enzyme here. Separate nucleus. Here separate enzyme sharing common subunit. Separate. So both are separate. Sorry. In single, here separate. Here also separate. Actually, initially I told that the type 3 also single, but not single one. The separate enzymes like a 2. The nucleus subunit structure, heterotrimer, different. These are homotrimers, same. These are also heterotrimer. Cofactor here, the ATPA cofactor, magnesium and SAM. Here only magnesium and magnesium plus SAM. These are cofactor. DNA cleavage. DNA cleavage requirement, 2 recognition site in any orientation, 2 recognition site because one recognition for methylation, single recognition site. Here 2 recognition site and orientation. Enzyme turnover number, no turnover number, first to 1. But turnover number present in endonuclease 2 and 3 and also 3 turnover number there. Turnover number means enzymatic turnover number within a minute one enzyme converts into how many products. If enzyme act on the one substrate after 1 minute, how many products are formed? The product number called as turnover number. It is specific to each and every enzymes. DNA translocation, first enzyme yes, and remaining two no. Site of methylation at recognition site. Both all the site of methylation at recognition, but variation is that within the same enzyme it lies or different enzyme it lies. That is same enzyme first and different enzyme remaining two. Cleavage pattern, some common restriction under given sticky ends. Mostly two restriction into two form sticky ends like this. Blunt ends, the same parallel cut, this form blunt ends. Application. A restriction into nucleases. In various applications related to genetic engineering, DNA is cleaved by using these restriction endonucleases. First of all, these are the mainly used in plasmid vectors during gene cloning and protein expression experiment. Insertions of the gene into plasmid. Insertion into gene into plasmid vectors. That is the first one. Restriction enzymes can also be used to distinguish gene alleles by specific recognizing single base change in DNA that is called SNPS. This is only possible if mutation alters the restriction site present in the allele. It is generally not occur. The restriction enzyme used for restriction fragment length polymer I will explain later. RFLP, HFLP, analyzed for identifying individual strains, particular species. So for identify individual strains for particular species. So this is about and polynucleotide phosphorylation not in syllabus. Phosphatase enzyme, right. Another enzyme in genetic engineering utilized that is phosphate enzyme. 1 minute. Phosphate catalases cleave the phosphate group from the substrate by using water molecule. Hydrolytic cleavage that is called hydrolytic cleavage. There is then from substrate water using water molecule. That is called hydrolytic cleavage, hydrolysis also. It is not reversible. After separating a phosphate group, never join. So the total opposite by kinase enzyme activity, this reaction, phosphatase enzyme reaction, opposite kinase and phosphorylase. What happened? Here actual phosphate catalyst cleavage phosphate for substrate by using water molecule. By using water molecule, cleave phosphate group from substrate. But what happened in kinase and phosphorylase activity? Add phosphate group to this substrate. On this basis, their activity, there are two types, phosphatase, acidic and alkali. First, for example, this is the phosphate group utilizing ATO, separate HBF and form H2OH, form H2OH. So this is schematic representation of hydrolytic cleavage of phosphate group. So this is phosphatase enzyme, which is done by phosphatase enzyme. And acidic phosphatase and alkaline phosphatase, that is the based on the pH level. pH between 3 and 6 that is acidic. pH about 10, that is the alkaline phosphatase. They're based on the chemical nature. Next one. Bacterial alkaline phosphatase, Gulf intestinal phosphatase. So let's see the other uh, alkaline phosphatases here. So here, uh, the kinases and phosphatases, common enzymes in modern day molecular biology laboratories. There are several sources of alkaline phosphatase. One is intestinal alkaline phosphatase, bacterial alkaline phosphatase, and the strip alkaline phosphatase, which is CIP we will discuss. The most frequent use of let's see. Most common utilizing is one that is CIP, cough intestine alkaline phosphatase. and T4 polynucleotide kinase. The both are modified the phosphorylation state to the 5' of DNA molecules. The mostly CIAP is most commonly used to remove 5' phosphate from the vector DNA to prevent the self-ligation during cloning. When the vector DNA is dephosphorylated, the insect DNA should not be dephosphorylated as 5S phosphate. Required for successful ligation reaction. Like that, the T4PNK, that is the T4 polynucleotide kinase, is routinely used to transfer phosphate from nucleotide triphosphate ATP to the 5' oligonucleotide to facilitate ligation. So mainly the 2,4-PNK has a 3' phosphatase activity that can be used to remove phosphate group from the 3 terminal of DNA and RNA. Although the both enzymes are most commonly used for cloning purpose, they have other activities and are also used for other type of studies. These other activities are listed further details. I will explain later. Next, applications of the Gulf intestine phosphatase catalyze the removal of 5' phosphatase group. Remember, 5' phosphatase group from either RNA or DNA. Self-ligation of the duplication can be prevented by this enzyme. Self-ligation, that means the same within the same DNA cannot bind each other. The enzyme action will cause the ME phosphorylated duplexes to be ligated to one strand and remain naked on the other. Next one. So given here the bacterial alkaline phosphatase, given the bacterial alkaline phosphate is a phosphomonostar and hydrolyze the 3' and 5' phosphate from nucleotide DNA and RNA. It more suitable remove the phosphate group before the labeling and remove phosphate from the vector where insect before the insertion for separation desired genes. These are the mostly used. This generally shows the optimum activity temperature 25 degrees centigrade. If you increase that, it can easily denature. It is bacterial alkaline phosphatase is a sensitive inorganic phosphatase. The presence of inorganic phosphatase activity may reduce. Shrimp alkaline SCAP phosphatase not given in our syllabus also. Next, methylase. Another enzyme, methylase. Methyl transferase or methylase catalyze the transfer of methyl group. Already discussed also this one. Methyl group to the substrate. The process transfer methyl group to its substrate is called methylation. Simple. The methylation is a common phenomenon in a DNA and protein structure. The methyl transferase uses reactive methyl group that bound to sulfur as adenosine methionine, which act as a methyl donor. Methylation normally occurs cytosine. This is important. Methylation normally occurs in cytosine residue in DNA sequence. In protein, methylation occurs nitrogen atom, the N terminus inside the chain protein. DNA methylation regulates, just discuss this one also, regulate genes and silent gene without changing DNA sequence. Epigenetic. That is called epigenetic regulation outside. In bacterial system, methylation plays major role in preventing their genome from degradation of restriction enzyme. Actually, restriction enzyme, where can found restriction enzyme presence in DNA? What is the function of restriction enzyme? Actually, restriction enzyme tend to be cut the DNA, but actually cut the genetic material. That is the function of restriction enzyme. These restriction enzyme found in bacteria, but within the same species, restriction there and also genome material also there. But the same species, why can't cut the same DNA, genetic material? If we separate this genetic enzyme, they can cut the DNA. But why can't cut the same genetic species? Because Methylation because of methylation, but it has the methylation process. Due to the methylation, it is prevented. Methylation can be three groups: M6A, M4C, M5C. So let's see here the methylase enzyme. So here, equal methylase activities. Methyl, add methyl group. Here we can observe methyl group. This one here also add by methyl group. So that is the methylase and any other extra information in our book. Let's see methylation in our book also. So we'll have to look through this, okay. And this methylation of DNA usually has a effect of protecting the DNA from the related restriction enzyme in order to do some restriction enzyme. Some restriction endonuclease will only cut at the recognized site of the DNA methylated. And still other restriction enzymes will cut the both methylated and non-methylated DNA. They recognize it. Example, BOM H1, one of the restriction endonucleases, and any other importance here. So these are the methylase enzyme activity. And next one, ligases. This is also important. Ligases enzymes important. They are very important. If asked an essay question, that easily write about the genetic engineering enzymes. Write about genetic molecular genetic engineering enzymes. Or if they want to ask individual ligase enzyme one and types of actual types of restriction enzyme basic and ligase one of the short answer. Just now we completed methylase. Methylase also one of the short answer. So what is ligase first? Ligase name itself ligase ligand formation phosphodiester bond attachment. Ligase means attachment. Phosphodiester bond where it attaches phosphodiester between the two deoxynucleotide residue in two strands. For example, our book given the not polymerases. Okay. Okay. Yes. So the ligase activity formation phosphodiester bond between two deoxynucleotide residue, two strands, where it is exactly require free hydroxyl group at 3 end, DNA chain and phosphate group at 5 end. 3 hydroxyl 5 end, phosphate group at 5 end. Other requires energy in the process. And it is joined by covalent bond. Creating phosphodiester bond between the 5' phosphate to 3 hydroxyl group. This is important. The bond formation 5' phosphate group to 3 hydroxyl group. The enzyme is active both blunt and sticky ends, but is more efficient on the latter because complementary ends can base pair, forming more stable substrate for the enzyme. For this action, for the ligase action requires what are requirements equal other bacteria and also whatever here. So other bacterial ligase NAD as energy donor. T4 DNA ligase uses ATP as a cofactor and also requires both magnesium ions and ATP for activity. The most common used ligase is the bacteriophage T4 DNA ligase. T4 this one. T4 DNA ligase. This is the most common one. So the DNA ligase are primarily responsible for joining gaps, joining process here important. So normal synthesis DNA, repairing the DNA. It has been exploited to no need. Just joining, okay. Joining Okazaki fragment, I will explain later. Okazaki fragment DNA repair recombination process, and the most widely used DNA ligase is T for bacteria. Ligase needs ATP as a cofactor mechanism. ATP, NADP react within ligase enzyme, form covalent enzyme AMP, linked amino group residue. AMP actives the phosphate group at the 5' end DNA molecule to be joined to 3' end of the hydroxy group. The final step is nucleophilics, 3 hydroxy already told you. So this is ligase. It requires already we can see here ATP required, magnesium 2 plus required, ATP convert into AMP. Here hydroxy group, OH group, hydroxy group to connect at the phosphate group. Here phosphate group. This ligase enzyme connected here. This here process. So joined. This both are joined now. 3 and 5 and joined. Well, application, the DNA ligase used in the cells join the Okazaki fragments in DNA replication process. In molecules cloning, ligase has been used routinely used in construction recombinant DNA. Following some of the example application ligase enzymes in molecular cloning, joining adapters, linker to blend. It's a simple. Here, first of all, Okazaki fragments. What is Okazaki fragment? Okazaki fragments means DNA replication. What is replication process? This DNA strand is separated and form as a new DNA strands. Blue color. Blue color. This is called replication process. But what happened while always one strand while synthesizing this DNA, one strand almost this is 5 and 3 and 5 and 3 and 5 and 3 and. So one is one direction, another one different direction. For example, here start 5 and 3 and here 5 and 3 and. What happened here? And take blue color. From this DNA polymerase help to synthesize the new strand to without to interrupt. What happened from this side, opposite side? Find here also find 3 end, but one pitch, one DNA strand. Here break. Since another DNA break, another DNA. This break occurs every pitches, every helix. So these fragments called Okazaki fragments. With help of this, I guess, for example, this 5 end opposite, it is 3 end. Again 5 and 3 end. Now this gap, 3 end gap, 5 end gap here, 3 end and 5 end, this both gap filled by ligase enzyme. Here also filled by ligase enzyme. Now these both are joined covalently. These both are joined covalently. So that is the best application. Next one. Polynucleotide kinase, no need. Polynucleotide given or not? Right, nucleases enzyme. Here given ribonucleases RNA enzyme. The nucleases that can catalyze the hydrolysis of ribonucleotide from the either single strand or double stranded RNA sequences. It is classified two types dependent on cleavage, that is endoribonucleases cleave internal bonds, exo cleave terminal bonds. Simple, like yeah, first one also. So RNA is important in RNA maturation process. Uh, this one RNA is a RNA is 2 both are plays important role in initial defense mechanism against RNA viral infection. Some common RNA enzymes are the ribonucleases A enzyme. It is the endoribonuclease that cleaves the specific single strand RNA at three end of the pyrimidine residue. The RNA is degraded into three phosphorylated mononucleotide C and U residue and oligonucleotide in the form of two 2 prime, 3 prime cyclic molecules. The optimal temperature for RNA is about 60 degrees centigrade. This RNA is can be inhibited by alkylation HIS12 and 19. It is used to remove the RNA contamination from the DNA sample. So this is about RNA. Nucleases. Methylase already completed. Now cloning vectors. Let's take the cloning vector. Just wait 1 minute. Next topic is cloning vectors. This is also very important. Cloning vectors in essay and short answer. Genetic engineering, cloning vectors, carrier for and DNA cloning. Now, cloning vectors are DNA molecules that act as a vehicles. Vectors means here the act as a vehicle for uh, carrying a foreign DNA fragment when inserted into the host cell. For example, uh, for example, this is the host cell. This is the one organism. From this, you separated the desired gene. This gene carried by one vehicle and carried towards to the host cell. So basically, one of the genome DNA, the DNA inserted with this desired gene and transferred into the another cell. So this is called gene cloning. So a cloning vector is a DNA molecule in which foreign DNA can be inserted. Foreign DNA. Foreign DNA can be inserted. This is one DNA. This is foreign DNA or desirable DNA, whatever it may be. And further capable replicating within the host cell and produce multiple clones of recombination DNA. That is the concept. This vector can be used for cloning to get a DNA copies in the fragment to obtain express in the cloned gene. Some characteristics of the vector, the most of the capable of autonomous replication and small in size, which can be convenient to take a desired gene and presents selectable markers of genes. The selectable markers, the selected marker gene, the genes permit selection of host cell with their recombinant DNA, those which do not bear recurrence. Example, AM selection genes some are AMPR, TETR. They are the different type of genes. Selectable marker genes. Presence of unique restriction site. Unique restriction site. That is important. It should have it should have restriction site to allow cleavage of specific sequence by specific restriction endonucleases. So to recognize the restriction endonuclease, it should be consistent with specific site. The unique restriction site should be present on the vector DNA molecule, either individual or as clusters. The cluster like polylinker. The polylinker multiple site. That means since there are many different restriction enzymes, it would be convenient to have wide range of restriction recognition site in the vector. It is allowed by the inserting a polylinker into the vector. The polylinker here, the E. coli complex bomb exchange. So it's utilized by polylinker sequence. And the vector easy purification, minimal amount of non-essential DNA. Now let's see the types of vectors, cloning vectors. Different types of cloning vectors are used for different types of cloning expressions, amplification. The vector is chosen according to the size and type of DNA to be cloned. Maybe smaller, larger, that is based on that. And it also depends on the host cell type used, prokaryotic and eukaryotic. Prokaryotic also different vectors and eukaryotic different vectors. First one, plasmid as a cloning vector. But the most common plasmid. Plasmid self-replicating, double-stranded, circular DNA molecule that are maintained in bacteria as a inadequate extra chromosomal entities. This one, this chromosomal, extra chromosome, this plasmid. Actually, the all bacteria genera have the natural plasmid. Almost all have the natural plasmid. Although they are not a typical essential for the bacterial survival, plasmid often carry genes that are advantageous under particular conditions also. That's why most of the bacteria are inconvenient to the pathogens. The plasmid can range in size from less than 1 kb to more than 500 kb. Each plasmid has an origin replication site inside the host cell because it need to be replicate. Plasmids are represented by 10 to 100 copies per host. Plasmid can carry DNA fragment already told you last time. The simplest cloning vector and the most widely used gene cloning are those based on the small bacterial plasmid. The plasmid has the ability to clone reasonably large subunit, large 100 base pair to 10 kbp pieces. Initially, naturally occurring plasmids were used as vectors, natural, not artificial. The natural occurring plasmids are PSC101, PS2124, Cola E1, and artificial PBR32. That is not given here. But remember PBR 322. So it is artificial. So however, the natural occurring unmodified or non-engineered plasmid often lacks several important features that are required for a high quality cloning vector. Therefore, the natural occurring plasmids cloning vectors have to be genetically modified. The modification include the size of the plasmids are reduced. If it is large in size, need to be reduced. Non-essential and toxin producing genes are deleted. Of course, in the plasmids also, some of the essential or non-essential toxin producing cells are there. First of all, we need to remove it, delete it, because if we selected as a vector toxic gene, what happened if the toxic gene inserted into host, the means the host would be killed. Selected marker genes are inserted, and addition of RE site restriction endonuclear site. One of the given PBR 322 vector was designed by Bolivar. Here the B for Bolivar Rodriguez, B for Bolivar Rodriguez, 1972. The size 4,361. The name PBR32 indicates the name PBR32. Lower P for plasmid, already told you this plasmid. B are Bolivar and Rodriguez, who developed the plasmid. 332 serial number. That is the serial. This is PBR32. Useful properties: small size. In PBR32, small size because just can be 10 kb below. And the presence of two antibiotic resistant genes, AMPR and tetracycline. If resistance, antibiotic presence, if they can kill other organisms. Here given this green color, tetracycline region. Here, green tetracycline resistance, yellow amplicycline. So two are present. They enable the selection of transferred cell from the non-transferred cells and recombinant from non-recombinant cells. And another useful property, a reasonable high copy number. Within the few minutes, they can develop 2,000 to 3,000 plasmid amplifications. Useful property can take up 2 kb of additional DNA. Pedigree, not important here. Important. Almost same characters in PUC vectors also derived from PBR32, developed at the University of California 1980. There are several vectors. PUC series includes PUC 8, 9, 18, 19, and so on. High copy cloning vector for replicating E. coli, like natural one. The PUC is suitable for blue white screening technique. The larger genes contain a cluster of unique restriction site ligation of new DNA into any one of these sites result in insertional inactivation of the genes and hence the loss of weak galactosidase activity. This is the key to the distinguished recombination plasmid one that contain an inserted piece of DNA from a non-recombinant plasmid that has new DNA, that has no new DNA. You see, actually in our book not given these are all. So viruses vector, before viruses vector, some other importance about this plasmid vector. The term first plasmid was first introduced by American molecular biologist Joshua Lederberg. First Lederberg. The types of plasmids also given. One way of the grouping plasmid is by their ability to transfer other bacteria. Conjugated plasmids, so-called transgenes, which perform the complex process conjugation. Non-conjugated plasmids incapable of initiating conjugation. Hence, they can only transfer it with assistance of conjugated plasmid by accident. An intermediate, an intermediate class of plasmid are mobilizable and carry only the subset of the genes required for transfer. They can parasitize conjugate to plasmid, transferring higher frequency only to presence. The plasmids are now being used to manipulate DNA and may possibly be a tool of carrying many diseases. There are different type of plasmids are given. Just the one F plasmid. F plasmid, R plasmid, coli plasmid. So they are the different type of plasmids. F plasmid, which contain trans genes, they capable of conjugation, transfer genetic material for touching. That means F plasmid present in this one bacteria. Here another bacteria. One bacteria have the F plasmid that have the pili. So this is the plasmid and this is genetic material. Whatever it is replicate, this plasmid replicate and this replication part migrated into the first of all here that is tube form as a like this one gene tube formation between the A and B. For example, this is the A bacterial cell, this is the B bacterial cell. So after replication of plasmid, this is converted into this one, migrated into the B cell. Genetic material not move, not transferred as it is. So later what happened? This is normal. Actually, initially this is only genetic material present. But apart from the genetic material, plasmid also migrated to that. So that is the F plasmid type. And resistance type, which contain genes that build resistance against antibiotic and plasmids. Plasmids also some sites restriction to the antibiotics. Polyplasmids, which contain the genes that code for determined products of bacteriocoins, protein that kill the bacteria. Mags of toxins, which are produce the toxins which kills the pathogens or bacteria. So these are the different types of plasmids. And next one, cosmid vectors. Virus vector, phase vector. First phase vector. So advantage of phase vector over plasmid vector, highly effective for the cloning of larger DNA fragments only, not only smaller and larger also, but plasmid vector only for small DNA. But this bacteriophage vector utilized for the larger one also. Screening of the recombinant DNA molecule is easier. The most widely used E. coli phase vector, lambda phase, M13 phase, lambda phase or M13 phase. Let's say lambda phase vector. The bacteriophage lambda, which infect the E. coli, has been widely used as a cloning vector. The lambda is well characterized virus with both lytic and lysogenic life cycle. Both are life cycle. Actually, this is not your part. Lytic cycle, lysogenic cycle. Lytic cycle means if the bacteria phase attack the cell, immediately the bacteria cell lyses. That is lytic cycle. But in lysogenic, it takes some period to separate up to divide up the bacteria by refrigerant. It takes some time. That is called lysogenic. Lambda DNA circularization within the bacterial cell and the DNA inside the phase particular site region. The size of lambda genome 4.8 kb, almost complex structure. A linear viral DNA, particularly here circular given both. At each end, there are complementary 12 base pair long overall that is called as CAS sequences. Coincident sequencing, coincident. Inside the E. coli cell, these pair up and the cohesive ends are ligated together by host enzyme, forming the circular version of the lambda DNA. This linear, due to the cohesive nature, they form as a circular DNA. And the lambda phase particular can accommodated up to 52 kb of DNA, up to 52 kb almost. But the plasmid DNA not carry about 20. If a 15 kb DNA is removed from the genome, then up 18 kb new DNA can be cloned. The middle region 15 kb, the lambda genome is non-essential and may be replaced within the approximately 18 kb foreign DNA. The two classes of lambda phase vector, insertion vector and replacement vector. What is insertion vector? In this type, vector large segment of the non-essential region is deleted and the two arms ligated together contain at least one unique restriction endonucleotide into which new DNA can be inserted. So insertion vector. Vector large segment in this large segment of the non-essential region. Non-essential region may be toxic expressing region, maybe one of the example toxic expression region. Actually, this toxic expression region not important. That's why we can remove it. And remove that region and two arms ligated together when, for example, is the plasmid. Sorry. For example, this region not wanted, toxic region, some unnecessary region, this one. You cut here from this. Now these two ends, this end and this end, inserted with desired gene, inserted with desired gene. Jointed desired gene insertion. That is the insertion vector. Vector. And next one. Here given also name insertion J, insertion vector, different limited scope of cloning large piece of DNA, insertion replacement vector, lambda insertion vector, new DNA insert pink separated or region and pink added like this. Same normal lambda DNA, 49 kb inserted here, previous ligand. Replacement vector. Next, replacement vector means in which the central stuffer fragment non-essential DNA is removed and replaced with the foreign DNA. Actually, in first, actually not replaced, only separated that one. And in first one, actually, I forgot one thing. In first one, actually, insert in this type vector, large segment of the non-essential region is deleted, and the two arms ligate together. That's it. But what happened here? Replacement vector in which central stuffer fragment, that means non-essential DNA is removed. They're deleted, but are removed and replaced by with the foreign DNA. The replacement vector can generally carry larger pieces of DNA than insertion vectors. Here, this is new replacement. This is completely stuffer fragment separated and replaced by new one. And this is about phase DNAs sequences and whatever other characters in phase DNA, whatever given in the bacteriophage vector given. L13 bacteriophages, lambda phase, we consider lambda phase, L13 lambda phase. Now the cosmate. The cosmate means simple two different two structure together. The cosmate, the hybrid of lambda phase and bacterial plasmid, both together lambda phase and bacterial phase plasmid and behave the both plasmid and the phase active. And now so far we completed plasmid and completed phase. If we both inserted, that called as a cosmid. Then actually the cosmid is a small multi-copy plasmid that carries lambda cos sites, origin site, MCS, AMPR, antibiotic, and can carry around 45 kb of DNA to be cloned. Enzyme that package the DNA into three phases, into the phase D protein. The code needed only the cause site and in vitro packaging occurs not only with lambda genome, but also with any molecule that carries cause site separated by 37 to 52 kilobase pair of DNA. It can easily pack it into phase at like a phase with cause site. They are able to replicate in the bacterial cell like plasmid, and origin of replication allow replicates in E. coli. Usually, cosmid size range from 5 to 7 kb, very less, and it can carry 45 kbp foreign DNA to be. Of course, it is minute in molecular weight, but it carry up to 45 kilobase pair foreign DNA. During the construction of the cosmid, the most of the lambda structure is deleted. Cast sites only remain, except cast, that is the active site, except cast that most of the undetected matter deleted. The modified structure enable phase heads to be stuffed with donor DNA. Example PJ P8. High transformation efficiency advantage, carry up to already told carry up to 45 kilobase per DNA fragment. Easy identification on the selective media, amplification and maintenance because it both properties, plasma property and phase plasma. Actually, M13 not discussed there. Same actually lambda phase discussed like that. This M14, M13 also. And because Plasmid features and uses. The plasmids predominantly plasmids with bacteria orientations, origin site, antibiotic selection and marker, and cloning site. And that means the selection of marker and also cloning site also. But they carry one or more recently two cause sites derived from the bacteriophage lambda. Uh, depending on the particular aim of the experimental board, host range cosmate, the settle cosmates or mammalian cosmates, they are available. The loading capacity of the cosmate varies depending on the size of the vector. Of course, it is depending on the size of vector, but usually lies around the 45 kilo base pairs. But the vector base pairs 4 to 5. Even if it is 4 to 5, it carry 40 to 45 kilo base pair. Cloning procedure involves the generation of two vector arms which are then joined at the foreign DNA. That's very interesting. The selection against the widespread cosmic DNA is simply done by a size exclusion. However, they always form colonies and not flood. They also the clone density is much lower with around 105, 106. In cosmic, the target DNA, the genome DNA to be cloned has to be cut into appropriate size range of restriction fragment. This is usually done by particular restriction followed by either size, fluctuations, and dephosphorylation to avoid chromosome scrambling, that is ligation and physically and linked fragment. Let's see disadvantages of the cosmic vector. The packaging enzyme fails to pack recombinant cosmate to phase reactor. If any one of two cause X is missing, that is the one disadvantage. Sometimes more than one recombinant cosmates join together, form larger DNA. If so, the packing enzyme fails to pack DNA into phase. So that this both are two disadvantages. 13. M13 reactorophage vector is a filamentous reactorophage E. coli and contains 7.2 kb long single stranded circular DNA. Long, not a double stranded, remember. Long single stranded circular DNA. M13 phase has been variously modified, give rise to M13 series cloning vector, which can be used cloning a wide variety of DNA fragments, particularly for the purpose of sequencing through strongest methods, redoxy chain termination. And then the cloning vector M13 series have Z gene complements gall host, giving blue colonies, but if foreign DNA segmented or inserted into polylinker site associated with Z genes, inactive lack Z genes, and no complementation is possible. Thus, a transformation won't be white or clear. Spots are obtained, thus permitting selection recombination M13 spots or M13 markers. Once the foreign DNA is cloned, M13 vector commercially available oligonucleotide primers are used for copying the insect in presence of deoxynucleotide, so that the fragment of different sizes known terminate produced, permitting the determination of the sequences. Reverse order, the restriction site in polylinker is present in pair of vector like M13, 8 and M13, MP9, permitting sequence from both the ends of double-stranded DNA molecule. The extension of the commercial primer or cloned fragment in M13 vector in the presence of radioactively labeled nucleotide will also allow generations of radioactive single-stranded probes. So this is about M13. And this is the 6,400 gene base for in 13 as can closed labeled genes in essentially for replication. 13 phase genome log Z gene, already discussed. Log Z gene is introduced into the intergenic sequences that near start log Z gene here given diagrammatically. M13 additional RE site restriction endonuclease site or restricted site are introduced into the log Z genes M13 to achieved by it is polylinker, this polylinker BHA. What's these are different type of polylinker mean different type of restriction endonucleases attack here. Advantage useful for producing single standard version only. Only 1.5 kb, very less Dalton, 1.5 kb Dalton. DNA can be efficiency clone, but more than 2, it is not used. It is only as it is single standard, it is only used for single standard DNA for several techniques such as DNA sequencing and in vitro mutagenesis. Phase mid. Phase mid is ideal vector of the combining of part of M13 genome and plasmid DNA. Lambda plasmid cosmid. M13 plasmid phase mid. Example PMBLA. This was made by combining regions M13 and PUC8 phase vector. Here the spots are AMPR and PUC8. Log Z genes with MCS from the PUC. Here also log Z genes play important role. Here 1300 base pair fragment from M13 single stranded DNA synthesizing region. Single stranded fragment up to kp in length. They are not just HC high capacity vector, whether it is given or not, look. Actually, in M30, not given. Just this high capacity vector, artificial chromosomes, need. Yeah, yeast artificial chromosome. Actually, this artificial chromosome introduction not important here. Actually, for large eukaryotic genome, researchers often begin with the vectors that can accept chromosome DNA insert very large size. So that is artificial, but here only one artificial genome. Yeast artificial genome, yeast artificial chromosome. First of artificial chromosome to be developed by David Burke, George Carley, and Meland Olson, 1987. Huge segment of DNA up to 2000 base pair, perhaps 2 million base pairs. Example PYC3. Essential for P where that plasmids into which a number of yeast genes have been inserted. It's only for artificial PBR32. Three genes are there, that is URA3, SUP4. The functions as the selectable marker. These are the used as a marker, selectable marker. SN is the centromere sequence of the chromosome. The telomere is proved by the two sequences called telomere TL. ORI is the bacterial origin replicating, and ORS is the yeast origin of replicating. ORI is the site of the origin, ARS site of the origin in yeast. A structure of the yeast artificial chromosome or PYAC3. Ag vector contain two telomere tel, one centromere cell, bacterial origin ORI, yeast origin ARS, selective marker TRPI, AR, URA3. So cloning procedure. The egg has two forms of circular form growing here in bacterial and linear form. Whether it is bacterial, this linear form and also circular form, two forms it can change. The circular form can be manipulated, can grow like any other plasmid in bacteria. So it has a bacterial origin, replication, and antibiotic resistance genes. In order to use this yeast, the circular form is isolated and linearized. But in bacteria, circulated okay. But it is they've used in a yeast cell, but it need to be linearized application. The cloning vehicle that propagate eukaryotic host cell can be routinely used 1600 kb fragment of 600 kb special type antigen DNA up to 400 kb length they can carry. Allow study of functioning and expression of the genes that very long. Under some circumstances, YEC can be propagated mammalian cells, enabling function analysis to be done in the organism in which gene normally resides. It is very important in the creation of gene libraries, gene library formation. So insert instability, not a stable insertion. Cloned DNA sometimes become rearranged by intramolecular recombination. When intramolecular recombinations are present, they cannot insert other desired gene. Another same such that bacterial artificial chromosome. YAC yeast artificial chromosome, BAC bacterial artificial chromosome. So BAC were first developed from the F plasmid, F type of plasmid in E. coli. Same, just compare with YAC. Small changes are there. All right. Thank you all.